Hi dear students, in the previous session we discussed about classification of bacteria and the mode of nutrition, on the basis of mode of nutrition. In this session we will discuss about the staining of bacterial cells and also some other concepts which are required to understand the nature and ultrastructure of a bacterial cell. Okay, we'll start the first one, staining of bacterial cell. So here staining refers to coloring or in terms it also called as dyeing. Dye ಅಂತಂದ್ರೆ ಏನಪ್ಪ ಕಲರ್ ಅಂತ ಕರಿತೀವಿ. ಕಲರಿಂಗ್ ಏಜೆಂಟ್ಸ್ ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಸೆಲ್ ನ ಕಲರ್ ಮಾಡ್ತೀವಿ. So that process is called as coloring or dyeing. Here some of the bacterial cells that you are going to learn is classified into gram positive and gram negative bacterial cells. Why we are going to classify them, or we are going to say those as gram positive and gram negative, and what is the basis for saying that gram positive and gram negative? Here is one solution for that. We can able to identify the cells on the basis of discovery by Hans Christian Gram. He is a scientist who worked on staining process for bacterial cells. Okay na? So here Hans Christian Gram worked on staining process for bacterial cell, and he used many chemicals to stain the cells. So in that, step 1, in first step we will find that collection of or culturing of bacterial cell. So if you take petri dish, and you can able to identify petri dish in your laboratories where it is spherical in shape, a glassware petri dish, in that you will add a nutritive media. So this is a nutritive media. What do you mean by nutritive media and how it will be prepared? It is made of a substance called as agar, and this agar is obtained from some algae, red algae in the extract ಮಾಡ್ತೇವೆ, and here this agar containing nutritive media, and this nutritive media which helps in growing of bacterial cells or microbial organisms. ಎಲ್ಲ microbes ಕೂಡ ನಾವು ಇಲ್ಲಿ grow ಮಾಡಬಹುದು. So this microbial cells are growing in the nutritive media, and we can add only few cells. We just add 5 bacterial cells. In next few days you can find that in a petri dish various number of cells are being grown. So in this petri dish you can able to find out the so many number of cells, and those cells are daughter cells of the bacteria which have added. Earlier we have added the bacterial cells, no? So those cells which give rise to a daughter cells, and from this we will take a bacterial suspension ಅಂತಂದ್ರೆ matter ಅಂತ ಹೇಳ್ತೇವೆ. So this suspension is going to be distributed, or we are going to make a layer of a bacterial suspension on a clean glass slide. So ishtu region ಗೆ ನಾವು ಏನು ಮಾಡ್ತಾ ಇದ್ದೀವಿ, bacterial cells na place ಮಾಡ್ತಾ ಇದ್ದೀವಿ. Okay na? So on this we are going to make a distribution of suspension which is taken from the nutritive media. Later we just heat this gently. Here there is a difference for gentle heating and boiling. When you are going to conduct experiments in our laboratories, you can able to conduct 2 types of heating process: gentle heating and warming. Gentle heating or warming, or otherwise boiling. You have to boil the water where there is a presence of bubbles. ನೀರು ತುಂಬ ಗುಳ್ಳೆಗಳು ಬರೋ ತನಕನೂ ಬಾಯ್ಲ್ ಮಾಡ್ತಾ ಇರ್ತೀರಾ. ಒಂದು ಇನ್ನೊಂದು ಏನಂತ ಹೇಳ್ತೀವಿ, ಜೆಂಟಲ್ ಹೀಟಿಂಗ್ ಆರ್ ಜಸ್ಟ್ ವಾರ್ಮಿಂಗ್, ಸ್ವಲ್ಪ ತಳನ ಬಿಸಿ ಮಾಡುವಂಥದ್ದು ಅಂತ ಕರಿತೇವೆ. ಓಕೆನಾ? ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಜೆಂಟಲ್ ಹೀಟಿಂಗ್. For what purpose we are doing this is to fix the bacterial cells to the glass slide. To fix the bacterial cells to glass slide. Next, in this, you just use the same glass slide is treated with crystal violet dye. Crystal violet dye. I am coloring this one. Crystal violet dye use ಮಾಡಿಕೊಂಡು color ಮಾಡ್ತಾ ಇದೀವಿ. The bacterial suspension which has been heat fixed is treated with crystal violet for 30 seconds. Right? So this is being treated with crystal violet for 30 seconds. And next we will rinse off it. Rinse off. What do you mean by rinsed off? That means if you consider this as a glass slide. So just you have to place in a slant position. So ಒಂದು glass slide ಹೀಗಿದೆ ಅಂದರೆ ಅದನ್ನ ಈ ತರ slant ಆಗಿ ಇಟ್ಕೋಬೇಕು, ಸ್ವಲ್ಪ vertical ಆಗಿ ಇಟ್ಕೋಬೇಕು. So that is called a slant position. Okay na? So after that you just pour water slowly and that make sure that you are going to rinse off the glass slide. That helps in removal of excess color or excess dyeing. ನೀವು ಹೆಚ್ಚು ಕಲರ್ ತಗೊಂಡಿರುತ್ತಲ್ಲ ಕಂಪ್ಲೀಟ್ ಗ್ಲಾಸ್ ಸ್ಲೈಡ್ ಅದು ವಾಶ್ ಔಟ್ ಆಗಿಬಿಡುತ್ತೆ. By using water you are going to rinsing a crystal violet dyed glass slide and that helps in fixing of cells with colored crystal violet. And then after you are going to treat the same slide with dilute iodine. Dilute iodine. The next step is treating with dilute iodine. After the treatment with dilute iodine, it is again treated with 95% alcohol. Here 95% alcohol is taken for treatment purpose. When you are treating the glass slide after the completion of rinsed off process with the dilute iodine, it will turns into deep blue color. Why I am writing all these colors? It will be asked for CET question after the treatment with crystal violet and rinsed off, which is the color obtained by the end of treatment with dilute iodine in grams staining method ante keltare. Okay na? That is deep blue color. After the observation made, that is deep blue color, again it is treated with 95% of alcohol. Right? So here this is the end of grams staining method. Okay? By this two types of cells are being observed on the same glass slide where few of the cells are having the same color. Deep blue color only will be present. Deep blue color containing cells will be there and those are called as gram positive cells, and few cells are also there which doesn't possess any color. No color or decolorization process is observed, and the type of cells are said to be gram negative cells. Gram positive and gram negative cells, and these are the type of bacteria which you are going to study in this session. So here the deep blue color containing gram positive cells example is Bacillus. All the groups which are coming in Bacillus are Clostridium. Clostridium botanicum ಅಂತ ಒಂದು ಸ್ಪೀಶೀಸ್ ಇದೆ. ಬ್ಯೂಟ್ರಿಕ್ ಆಸಿಡ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡ್ತೇವೆ. ಕೆಮಿಸ್ಟ್ರಿಯಲ್ಲಿ ಬ್ಯೂಟ್ರಿಕ್ ಆಸಿಡ್ ಅಂತ ಯೂಸ್ ಮಾಡ್ತೀರಾ ನೀವು. Large scale preparation of butyric acid is done by using Clostridium species of Clostridium botanicum. Okay, and no color is observed. Our decolorization process has been taken place, and those type of cells are called as gram negative cells. And example is Vibrio. A small extension, thread-like extension has been observed in spiral-shaped bacterial cells. That is Vibrio. Cholera is one best example of gram-negative cell. And another one is Streptomyces. 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 So these are the two examples for gram-negative bacterial cell, and Bacillus and Clostridium are the two examples for gram-positive cells. So this is about the staining method for bacterial cells. You just summarize this one. Staining of bacterial cell is nothing but coloring process, where you can find many different type of bacteria. In that you can able to identify gram positive and gram negative, and how it will be done, and how would we are going to identify this is a gram positive and this is a gram negative cell, only by the contribution or the work done by Hans Christian Gram. He conducted an experiment to find out the gram positive and gram negative cells. First, they have introduced the bacterial cells to the nutritive media, which helps in growing of the bacterial cells, either by binary fission or by spore formation. And later, after few days, we will get the number of cells in the nutritive media. We are going to take a suspension of the bacterial cell on a clean glass slide, and it is going to be gently heated, and that process helps in fixing of the bacterial cell to the glass slide. Later, it is treated with crystal violet dye for 30 seconds, which makes the cells to turn into crystal violet color. And then it is rinsed off with water by positioning slantly toward the glass slide, and then again it is treated with dilute iodine, which gives deep blue color. And later 95% of alcohol is treatment. The treatment with 95% alcohol will finally gives us two types of cells. One is deep blue color containing positive cells. Example is Bacillus and Clostridium. The next is no color is obtained, or the cells of deep blue colored cells have been completely changed their color, or they will lost their color, and those are called as no color containing cells or decolorized cells, and they are also called as gram negative cells. Example, Vibrio cholera or the Streptomyces group of organisms. Right? So this is about the staining method of bacterial cells. So write down the same thing. You will going to face few questions on this in your CET or also in next year syllabus. Okay, na? So I will drop this one. Next, we will discuss about the ultrastructure of a bacterial cell. Very most important concept is going to be studied in this session, and in the next session we will find out the few more concepts regarding the ultrastructure of a bacterial cell. In this session we will take up the first concept, that is capsule or slime layer. Here you can find that the bacterial cells which may be present in a colonies. They may be present in a colonies. They may be separate single also, or they may be filamentous also, where there is a presence of continuous addition of cells in a ladder-like structure, and that is called as filamentous structure of a bacterial cell. These different type of bacteria are going to be observed, and on the layer of outer layer of bacterial cell colony you can find that there is a presence of a slimy layer, or it is also called as mucilaginous sheath. Right? So this mucilaginous sheath is nothing but capsule or slime layer. So this is called as mucilaginous sheath. And this mucilaginous sheath is made of two components. One is polysaccharide. That is called as slime layer, and second one is a component of polysaccharide and also along with this polypeptide. So in the next year ಜೆನೆಟಿಕ್ಸ್ ಅಲ್ಲಿ ನೀವು ಮತ್ತೆ ಮಾಲಿಕ್ಯುಲರ್ ಬಯಾಲಜಿನಲ್ಲಿ ಈ ಕಾನ್ಸೆಪ್ಟ್ ಮೇಲೆ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್ ಒಂದು ಕ್ವಶನ್ ಬರುತ್ತೆ, ಟ್ರಾನ್ಸ್ಫಾರ್ಮಿಂಗ್ ಪ್ರಿನ್ಸಿಪಲ್ ಆಫ್ ಗ್ರಿಫ್ಟ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅಂತೇಳಿ. ಓಕೆನಾ? ಸೊ ಅಲ್ಲಿ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್, ಇವಾಗ ಬೇಡ ಅಂತ ಬಿಟ್ಟು ಬಿಡಬೇಡಿ. ಸೊ ಹಿಯರ್ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಅ ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಮೇಡ್ ಸ್ಲೈಮ್ ಲೇಯರ್, ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಪ್ಲಸ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಮೇಡ್ ಕ್ಯಾಪ್ಸೂಲ್. ಓಕೆ, so this polysaccharide made slime layer is non-virulent, is also called as non-pathogenic. And they can be lysed. Lysed ಅಂತಂದ್ರೆ ಬೇರೆ ಮೀನಿಂಗ್ ಬರುತ್ತೆ, LYSED. This means breaking of cells. ಬ್ರೇಕ್ ಮಾಡೋ ಪ್ರೋಸೆಸ್ನ ಲೈಸ್ಡ್ ಅಂತೇಳಿ ಕರಿತೇವೆ ಬಯಾಲಜಿನಲ್ಲಿ. ಓಕೆನಾ? ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಬ್ರೇಕಿಂಗ್. By the activity of immune cells or maybe the activity of antibiotics will directly destroys or vanishes the bacterial cells by lysis process. This is also called as lysis. Okay na? Here, if we have any bacterial diseases, either by food poisoning or by taking of contaminated water, the bacterial cell will enter to our body and that shows some symptoms. If the symptoms has been observed by a doctor or a physician, he just suggests you that to take an antibiotic. Against the bacterial action, we are going to take out some drugs. And the drugs which we are taking will make a lysis of bacterial cells. Like example, slime layer containing bacterial cells, and that become non-virulent and non-pathogenic. Pathogen ಅಂದ್ರೆ meaning already ನಿಮಗೆ ಗೊತ್ತಾಗಿದೆ. Disease causing organisms are called as pathogens. Okay, and this type of bacterial cells are called R strains. And later one more type, a polysaccharide and polypeptide composition containing capsulated bacterial cells also there, which are virulent and pathogenic. And these are S strain or smooth strain containing. If we take any antibiotic, that will not work properly, or they will resist over the antibiotics, or the immune cells of our body will not respond properly to these type of bacterial cells. So that's why the symptoms will become severe to the bacterial diseases, and those type of disease caused organisms are capsulated bacterial cells. ಒಂದು ಸ್ಲೈಮ್ ಲೇಯರು ಅವು ಈಸಿಯಾಗಿ ವ್ಯಾನಿಷ್ ಆಗ್ತವೆ ಆಂಟಿಬಯೋಟಿಕ್ಸ್ನ ತಗೊಂಡ್ರೆ. ನೆಕ್ಸ್ಟು ಕ್ಯಾಪ್ಸೂಲ್ ಕಂಟೈನಿಂಗ್, ದೇ ವಿಲ್ ನಾಟ್ ಗೆಟ್ ವ್ಯಾನಿಶ್ಡ್ ಈಸಿಲಿ ಆ್ಯಂಡ್ ದೇ ರಿಕ್ವೈರ್ ಲಿಟಲ್ ಮೋರ್ ಎನರ್ಜಿ ಟು ಲೈಸಿಸ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ಸ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಸೊ ಹಿಯರ್ ಯು ಕೇಮ್ ಟು ನೋ ದಟ್ polysaccharide containing slime layer which are non-virulent and non-pathogenic and are called as R strains. And some other also there, polysaccharide plus polypeptide containing capsular cells which are virulent and pathogenic. They are also called as S strains. ಅರ್ಥ ಆಗಿದೆಯಲ್ಲ? So you will able to identify two difficulties in understanding the polysaccharide and polypeptide. When it comes to a single block, ಇಲ್ಲಿ ಒಂದು ಇಟ್ಟಿಗೆ ಇದೆ, ಇದನ್ನ ಸಿಂಗಲ್ ಅಂತ ಕರೆಯುವಾಗ ಕೆಮಿಸ್ಟ್ರಿಯಲ್ಲಿ ನಿಮ್ಗೆ ಹೇಳ್ಕೊಟ್ಟಿರ್ತಾರೆ ಮಾನೋ ಅಂತ ಹೇಳ್ತೇವೆ. ಓಕೆನಾ? ನೆಕ್ಸ್ಟ್ ಹೀಗೆ ಒಂದು ಸೆಲ್ ಇದೆ, ಅಲ್ಲಿ ಎರಡು ಇರುತ್ತೆ, ಬ್ಲಾಕ್ಸ್ ಎರಡು ಇರುತ್ತೆ. ಸೊ ಅವಾಗ ಏನಾಯ್ತದು ಡೈ ಆಯಿತು. ಹೀಗೆ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಆ್ಯಡ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಆ್ಯಂಡ್ ದ ನಂಬರ್ ವಿಲ್ ಬಿ ನ್ಯೂಮರಸ್, ತುಂಬ ಇದೆ ಅಂತ ಅಂದ್ಕೊಂಡಾಗ ಅದನ್ನು ಪಾಲಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಸ್ಯಾಕರೈಡ್ ಅಂತಂದರೆ ಮೀನಿಂಗು ಶುಗರ್ ಅಂತ ಹೇಳಿ, ಓಕೆನಾ. ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಕಾಂಪೋನೆಂಟ್, ದಿಸ್ ಇಸ್ ಅ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಕಾಂಪೋನೆಂಟ್. So there is a presence of many number of sugar molecules that leads to the formation of a complex carbohydrate that is called as polysaccharide. In some organisms like Bacillus anthracis. So this is a one bacterial cell which is rod shaped, which contains the glutamic acid containing slime layer. So ಇಲ್ಲಿ ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಯಾವುದು ಅಂತ ಕೇಳಿದ್ರೆ ಬ್ಯಾಸಿಲಸ್ ಆಂಥ್ರಾಸಿಸ್ ಅಲ್ಲಿ ಡಿ ಗ್ಲೂಟಾಮಿಕ್ ಆಸಿಡ್ ಆಗುತ್ತೆ. ಇದೊಂದು CET ಕ್ವಶನ್ ಬಂದಿದೆ ಆಲ್ರೆಡಿ ನಿಮಗೆ, ಓಕೆನಾ. ಸೊ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಅಂಡ್ ದ ಸೇಮ್ ಥಿಂಗ್ ಫಾರ್ ಪಾಲಿಪೆಪ್ಟೈಡ್. ಸೊ ಯು ನೋ ದಟ್ poly means many, ಓಕೆನಾ. ಸೊ here peptide means a protein unit. ಪ್ರೋಟೀನ್ ಅಂತಂದರೆ ನಿಮಗೆ ಗೊತ್ತಿರುತ್ತೆ, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್, ಪ್ರೋಟೀನ್ಸ್ ಆ್ಯಂಡ್ ಲಿಪಿಡ್ಸ್ ಅಂತೇಳಿ ಕ್ಲಾಸಿಫೈ ಮಾಡಿರ್ತೀರ, ಓಕೆನಾ. ಸೊ ಇಲ್ಲಿ ಮೆನಿ ನಂಬರ್ ಆಫ್ ಪ್ರೋಟೀನ್ಸ್ ವಿಲ್ ಮೇಕ್ಸ್ ಅ ಪಾಲಿಪೆಪ್ಟೈಡ್, ಓಕೆ. ಸೊ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದಿ ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಆ್ಯಂಡ್ ಪಾಲಿಪೆಪ್ಟೈಡ್. ಅರ್ಥ ಆಗಿದೆಯಲ್ಲ ಇದಿಷ್ಟು? ಕ್ಯಾಪ್ಸೂಲ್ ಆ್ಯಂಡ್ ಸ್ಲೈಮ್ ಲೇಯರ್. ಅಂಡರ್ಸ್ಟುಡ್? ನೆಕ್ಸ್ಟ್ we will discuss about the ultrastructure, other parts of a cell and cell organelles, okay. Thank you dear students.